I just start, okay. All right, so I'll talk about, so last lecture I talked about is about, you know, the mechanics of microtubule or filamentous bundles. And what we'll do now is we'll drive these bundles out of equilibrium. They'll be energy consuming, and you'll see they'll organize into a variety of interesting patterns that are intrinsically far from equilibrium. And so this work was done by primarily these three students. Tim really started it all, and Steve and Dan are working on it right now. And for part of the experiments, we collaborated with Andreas, who has a nice technology of encapsulating things into lipid vesicles and things along these. And so we combined our materials with his technique, and we got interesting results. All right, so it's okay. So you know, so if you look into a living world, you'll see that living organisms do a bunch of very interesting things. If you go into a pond, maybe even here in a local pond, and if you look, just, you know, you'll see in the pond, you'll see a bunch of, you know, ciliates, protists that look like this, that swim around, and these things are actually covered with dense hair of beating cilia that propel these things forward. And so a bunch of living organisms can swim. Another kind of ubiquitous property of living organism is that they can divide, right? So here you have a cell, it undergoes mitosis, and you get two daughter cells. Besides swimming, you know, a lot of cells have the ability to crawl, and so this is a movie that I think was taken in 1950s, where you have this neutrophil that is chasing a bacterium, and it finally catches up with it and eats it for lunch. And then, you know, and this is a final example, I guess, that's most relevant to our work, is a cytoplasmic streaming. So this is a very large oocyte, that means an egg of a fruit fly, Drosophila. Okay, and so this is millimeters in size, or a millimeter in size, and these bright spots that you see here is the food that's required for oocyte to develop and divide. And so if you were to rely on a diffusion to transport this food and uniformly steer it across the cellular cytoplasm, that would take a very, very long time. And so what nature has developed is this phenomenon of cytoplasmic streaming. And if you saw this phenomenon of cytoplasmic streaming is, you know, just enhanced mixing, right? So this thing spontaneously mixes itself. And so if you want to do this, you know, for your coffee, you just stir your coffee by putting a spoon in there and inputting energy on a macroscopic scale. But you don't have that option. A cell doesn't have that option, right? It can't input energy on a macroscopic scale, so you produce this material that self-stirs by inputting energy at microscopic scale. So it's very different. And so this is real biology, right? And I'm not really a biologist, and I don't really want to study these details of these processes, but these are interesting functionalities, and what we would want to do is we want to make materials that have functionalities that are described here, right? So we want to make highly simplified materials, start from a few basic building blocks, and ask questions, can we build materials that look like this? And something that's obvious is that these materials are far from equilibrium, or these organisms. So they inherently need ATP to drive these processes. If you don't have ATP, you would never get this kind of dynamics, right? And so, and the other thing that I would like to say is that these materials are driven by very simple building blocks, right? So all of them are driven by molecular motors and filamentous bundles, and so we want to start putting these materials in a simplified manner and see whether we can recreate this sort of materials. So what are my basic building blocks? One of the basic building blocks is what I talked about yesterday is microtubules. So these are, again, stabilized and they don't undergo depletion interaction. The other building block is this depletion interaction, okay? And so I add a depletant and this induces the bundling, and that's what I discussed last time. And so I only add one more component to this, okay? So yesterday I discussed the mechanics of these bundles, and now we add just one more component, and that's kinesin molecular motors, okay? And so kinesins are proteins, and they have this incredible ability to hydrolyze ATP, and when they hydrolyze ATP, every time they hydrolyze ATP, they take an 8 nanometer step along a microtubule, okay? And so this is 8 nanometers, these two dimers. And so it's a machine that converts chemical energy into mechanical motion. And so that's illustrated in this cartoon, right? So, you know, I don't think this is really real, but you get ATP, you hydrolyze ATP, you take a step, you release ATP, and you just keep walking. And on average, you take about, 100 steps, and at that point you dissociate, okay? Unbind from your trap. And so we're going to combine these three building blocks, and you'll see that you get interesting emergent properties. And so, like actin, microtubules are polar structures. That means that they have a plus end and a minus end. And you know, this is just my ubiquitous bundle that I showed you yesterday. You have a polyethylene glycol that holds this bundle together, and let's assume that they have equal polarity. That means that they both point in the same polarity. And so I add these motors, and again, you get this passive diffusion, but what we do now is we add motors. And we don't add one motor, because one motor just takes simple steps, right? What we need to do is we need to make a cluster of motors. So this is one kinesin that's labeled with this blue molecule that's called a biotin, and this is another kinesin that's labeled with biotin. And now you have another molecule that you isolate from nature, and it's called streptavidin, and streptavidin in principle has 4 binding sites for biotin. So if you just mix this biotin labeled motors with streptavidin, you will get a cluster of motors, right? That these clusters might have from 2 to 4 motors or even 1, right? And so this is a multi-motor cluster, and that's our basic unit, building block. If you take this cluster and you add it to microtubules, this is what's going to happen, right? Is this motor is going to walk along the microtubule towards the plus end, and this motor walks along this microtubule towards the plus end, right? And so the motors simply walk along, and they fall off. So nothing interesting happens yet. Now, it's equally, when I make this bundle, it's equally likely that the filaments will have same polarity or the opposite polarity. If they have the opposite polarity, what's going to happen? That means that the plus motor is here and this plus end is here. This motor is going to walk along this direction, and this motor is going to walk along this direction, right? And as these motors are walking along, they'll push the microtubules apart, okay? And the other thing that I should say is that by having this bundle geometry, you greatly enhance how long the clusters stay bound to the microtubule. On average, a single motor will walk 1 micron and unbind, but if it walks a micron and it unbinds, it's always bound to the microtubule with the other side, right? And so when it unbinds, it can again rebind because there's a lot of binding sites. If the microtubules were isotropic, the effective sliding of the filaments would be much less efficient. And so, okay, so what you should remember is polar microtubules, no interfilament sliding. Antipolar microtubules, you get this interfilament sliding. And so what we do is we take these filaments and we confine them in a quasi 2-dimensional suspension. And that's shown over here, okay? So here you have a bundle of microtubules. There's polyethylene glycol all around, and you have molecular motors that come to the microtubule, they bind and they drive them apart. By themselves, these bundles don't do any dynamics. That means that they are polarity sorted, okay? But if you just let them diffuse around, what's going to happen is initially you saw that they bound because of the depletion interaction, and they actively slid past one another. And then they rotate, so then this thing rotates by 180 degrees, and it rebinds. And when it rebinds, it stays bound to the microtubule, and the microtubule doesn't slide. So what we presume is happening, we don't know what a plus end or a minus end is, but initially you bound in antipolar sense, you are slid apart, and then you rotate, and then you bound with the same polarity, and the thing stays uniform, okay? And so you get these recombination events, and so, you know, isolated microtubules are static, which probably indicates that they're locally polarity sorted, but you get this bundle recombination that reinitializes local polarity sorting, okay? And so we just want to increase, you know, so you can watch this and you can study it, and there's interesting things that you can tell about this. But what we want to do is we want to increase the recombination efficiency, right? And the way we do that is we just increase the concentration of the filaments. And so you increase the concentration by an order of magnitude, and you get your sample, and the sample does something very interesting, right? Instead of just being a classical gel, it becomes unstable and develops this large-scale patterns.
Does this depend on how big the sample is compared to whatever the activity source?
I mean, you would think that it would stop when the sample is too small or when it's too big. I mean, we've made 1 ml samples like this.
When it's very small.
When it's very small, then you know you get them. You know, you can confine them in emulsions, and that's the second part of my talk, right? Because then you get this things become unstable, and they go to the surfaces because they're below the prop. There's a certain characteristic length scale here that's 100 microns, and so if you confine them below 100 microns, things will go to the surfaces. I'll describe that in a second part of the talk.
What's the initial state you start from?
I just mix it. You know, I just mix these 4 components, and I put it in the sample chamber, and I let it run, and I go quickly under microscope, and it takes me 2 minutes maybe to, not me, my students, right?
My question was, if you had started with something which is all aligned.
Yeah, I will show you that in the next slide. Yes. 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 Yeah.
So for small systems, you would just have a nematic that wants to align without doesn't want to spontaneously break the order.
Well, this is isotropic. This is a bulk 3-dimensional gel. I'll zoom in and I'll show you in a few slides. So this is interesting because again, it looks turbulent-like, but it's not driven at macroscopic length scales, but it's driven with this microscopic motors. Yes.
Understood what the geometry is when you say quasi-2D?
No, this is maybe 100 micron stick. So, but I can make it 10 micron stick. I can make it whatever. It's very robust, right? So, am I doing the beer thing as well, or? I just got paid, so I have, I have to spend all this money, so.
Will help.
So this is perhaps we start with a better aligned state. So what we do here is we shear align these things. So we just put it in a shear cell. We shear align. They are all initially aligned along this direction. And you'll see that you develop this instability that propagates in the direction perpendicular to the initial alignment, okay? And so this, I think, can be understood fairly easily, is these bundles are extensile, right? So the motors come, they might be contractile initially, but at the end they're always extensile, and they're most likely to bind with maximum overlap. So that's why that breaks the symmetry between contractility and extensility. So you have these extensile bundles, and let's assume that they're initially all aligned along the same direction. And then, of course, you'll get some fluctuations at some point. And so if you have a small fluctuation, what's going to happen is that you're going to this bundle is extensile, this bundle is extensile. The forces add up here in this direction, and the forces add up here in this direction, right? So what's going to happen is that you extend more, this initial fluctuation in bend is going to grow in amplitude, okay? And so this is an unusual material, right? If I usually bend a material and bend it, it wants to become straight. Here, if you have something that has an initial bend, it grows in amplitude. And so that's just typical of non-equilibrium things, I think.
Isn't there a sense that would be straight to like a bending?
Yeah, there's elasticity that you're fighting against, right? So,
But then that means that this modulation that would lead to the instability
yeah.
will be able to take over only if it's larger than the wavelength.
Yeah, so there's certainly a wavelength that we can measure, right?
And so below that wavelength, this will be suppressed. Because the tension between...
There is a length scale. There is a length scale.
Yeah, there's a certainly length scale selection, and that's related to this buckling instability. It's not unstable at all wavelengths. And so I think this was first described in this paper. And so these are very robust materials, right? So this is 1,500 microns, and so you can see this large scale patterns. And so these things are, experimental aggregates or pieces of crud. And so what you'll see is this is probably 20, 30 microns by itself, right? And so it's 20, 30 microns. And so if this thing was just sitting in a suspension, it would be like a brick. It would just be sitting at the bottom of your suspension. And this maybe takes 10 minutes, and you can see that this active gel actively transports this non-Brownian particle by hundreds of microns very effectively. So in this way, it's very similar to, to the cellular cytoplasm that I showed you, only that, we have 3 components, and the cellular cytoplasm has hundreds of components, and we can actually tune things and do systematic experiments. We can do physics with this kind of material.
This is real time?
It's probably sped up. These things move up to 10 microns per second. Okay, so it's probably sped up 10 times. And so what are the properties of these active gels? One is, this enhanced transport and mixing, and it's very robust. And the other thing that's very important that I should mention is that, this is all fed by ATP, right? So you put ATP, and that powers the molecular motors. Now, if I just put ATP and I start consuming ATP, that would make essentially making steady state samples impossible, right? Because ATP would continuously decline in concentrations, and the motors would continuously slow down. And so what you can do is you can borrow the tricks from cells, right? So the cell has a storage of ATP through, when you run a marathon, 100 meters, what you do is you have stored ATP, and you regenerate that ATP through certain biochemical cycles. And so we can just borrow those biochemistry and engineer it in our system to continuously regenerate ATP. And so these things will last for, multiple days, up to 2 or 3 days. And at that point, the proteins start to degenerate because we have to run our things at 37 degrees. There's actually microtubules that you can isolate from fish that are stable at 4 degrees. And so we want to really go and cool these things down that you can, in that case, I think you could run it for weeks. And so the questions I want to ask is, what are the macroscopic properties of these active gels? And, what is the microscopic dynamics, okay? So what's actually happening to these bundles? And so here is, if you zoom into this active gel, this is what you see, okay? These are your individual bundles now, and this is a time sequence. So you'll see 2 bundles here, and they merge because of the adhesive interactions, the depletion interactions. And so you'll see here, they merge, and as they merge here, this merging point increases, right? And as they merge, they are equally likely to have the same polarity or the opposite polarity. If they have the opposite polarity, they'll start extending and polarity sorting. But now they are in a gel, so if a single filament bundle extends, they can just simply extend. But since they're clamped at the ends, what's going to happen is that they're going to buckle at certain wavelength. I think, and that's the main wavelength that determines the behavior of the system. So you can see this point over here grows and buckles, and at some point the curvature becomes so large that the thing fragments into pieces. And now I have new microtubule bundles that can merge with the system, and you go through the cycles of bundles merging, polarity sorting, extending, buckling, fracturing, and the thing wants to polarity sort, but it just never gets to polarity sort. Okay, and so it's eternally unhappy. And so you see that in this movie, right? So here you have a bundle, it merges, it extends, it buckles, it merges again, and this cycle goes on and on and on for essentially as long as you have enough fuel. What's important to mention is that this length scale is 100 microns. Our single microtubule is 1 micron long, right? So what you're breaking when you fracture this thing is you're not breaking individual microtubules, you're really breaking this composite microtubule bundle that's held together with very soft depletion forces. And so that's something, that's why we started studying those frictional properties, because we want to understand what is the buckling behavior of a composite bundle instead of a single beam that's been studied extensively. And so what we are setting up to do now is what we are very interested in is actually tracking all individual bundles. And so we are doing this in collaboration with Dan Blair. So he has this very nice confocal microscopy that's set up on a rheometer, and you have this gel, and you can ask, well, is this gel, is it a liquid or is it a gel, or what is its rheology? And so, so you can do at low ATP, the system is slow enough that you can actually track the three-dimensional topology of this network. And so this is a three-dimensional reconstruction, and something I will not talk about. But if you do rheology of this thing, it just looks like a gel. So that's very surprising to us, right? G prime and G double prime are just flat. And when you look at this thing, it really looks like a liquid. So that's something that we completely don't understand, but we have extensive measurements on that. Okay, so what you can do now is you can say, what are the macroscopic properties of this material? So they're perfectly tunable, right? And so we tune how fast the motors walk by tuning the ATP concentration. 0 ATP concentration, it's just a classical gel. Is everything? Oh, okay. And so these beads are really trapped in this gel, and they don't do too much, right? If I increase the ATP concentration, here you can see that the beads start slowly to move, and at saturating ATP, the beads move like crazy. These beads are not attached to the microtubule network, they're just diffusing around, and they're being advected by the flow that's generated by the background network. And so you can measure this mean square displacement as a function of time that you've seen multiple times here. And again, let me remind you that a mean square displacement that increases linearly with time is representative of normal diffusion, right? For 0 ATP, that's the blue curve. This is slope 1. So if you measure something like this, that would indicate normal diffusion. For low ATP, what you see is that this thing really behaves subdiffusively, right? So this indicates that the beads are trapped within your network, and this is a characteristic. If you put your beads in any gel, you'll measure a behavior like this. If you start increasing ATP concentration, you see that the transport becomes super diffusive. That means faster than 1, and that is only possible in systems that are actively driven. And then at the highest ATP concentration, what happens is that things scale with mean square displacement as time squared, right? And that means that I'm essentially just moving ballistically, right? I'm not really turning around, I'm just moving along the straight line. It should probably turn over at some point because they don't really move on the straight line, but we don't have the ability to track the beads for a sufficiently long time. And so this kind of nicely illustrates what is the tunability of the system, right? We can start from something that's really conventional gel, and we can increase the concentration. People have done these measurements on bacteria-like suspension, right? But in bacteria, you can't do such controlled experiments because bacteria, you can't tune their swimming velocity. They divide, they die, and things like that. Well, this will just keep going for a long time. questions so far? No? Students? The other thing that you can measure is you can measure the temporal dynamics of this flows, right? So so here is at 0 ATP concentration, nothing much happens. At 35 micromolar, the dynamics behaves like this, and this is at saturating ATP concentration. And so what you can measure is you can measure the velocity-velocity correlation profile, right? So what this tells me is if I'm a particle sitting at this point, how correlated I am with a particle sitting at distance r apart. Okay, and then you just normalize it by the mean velocity. And so you'll see, so this is the spatial dimension, and this is how correlated things are. So it says that anything within 20 or 30 microns is essentially moving together, and then this correlation function drops down, and it becomes, it becomes slightly anti-correlated, and then things above few hundred microns are anti-correlated. And so this tells you that there is and things actually scale, well, reasonably well at different ATP concentrations, right? So I can renormalize the speed, and there's a characteristic length scale, and this characteristic length scale is about 100 microns. And we believe that this is essentially determined by the buckling length scale of a microtubule. Okay, so that's the length scale of, yes? This is the velocity of a bead, or you can also do PIV, and you can track the gel, and they exactly fall on top of one another. But we made sure that the beads do not stick to the gels, because they're coated with.
Because they need the correlation of the gel itself.
Yes.
The equation length.
And the beads is the same. They're exactly the same.
That's not set by just the incompressibility of the thing and the thickness of the cell.
Say that again?
If you look at velocity correlation,
Yes.
the closed box. Yeah, but if you have an
Yeah, okay.
of the thing, you have an anti-correlation
Yeah.
that shows up at the box size.
No, this is much, much smaller than the box size.
Okay.
We can make these things. That we've also checked. We've gone up to 500 microns, I believe. Okay, so what I've told you so far is, I've started with I've showed you so far is we have this assembly of, we started with the simple extensile microtubule bundles, right? And at high bundle concentration, they make these isotropic gels that are characterized by spontaneous internal flows, enhanced transport and mixing, okay? And so what, I think what's interesting is if you confine these gels into a tube, for example, they'll exhibit very interesting things that we're just starting to study. So this is maybe a 100 micron tube that's shown over here. And so what you'll see is we haven't put the beads in here, and this is just a linear tube, right? And so you'll see that the edges, all the microtubules are spontaneously transported in this direction, right? But I can't just, yes?
If you had no ATP, would this be an isotropic system? Or is
This would be an isotropic cross-linked gel.
So there wouldn't be any nematic order?
There would be no nematic order, yes.
But eventually, if the concentration high enough, would these things...
that I'll quickly jump to. They don't equilibrate, right? They are gel-like substance, and at high concentration, they are just in a non-equilibrium gel-like state. And so you can't make a nematic out of microtubules because, you just get a birefringent gel that doesn't have any fluctuations, is not really a nematic. But so I think this movie is interesting because it shows you that, once you confine these things, you'll get, unconfined things will exhibit this large-scale chaotic flows. By confining them, you can actually channel this flow, and you can get a material that spontaneously flows in one direction. So here it can't flow in one direction because there's an end of this end, but on the outside you set this flow, and if you follow this material, on the outside it goes in this direction, but if you look in the center, the flow comes back in this direction. So you set this large-scale patterns, and so what we're trying to do is engineer these things in a circular geometry and see whether you would get something that really flows and generates, a macroscopic force. Yes?
Back to your rheology experiment.
Yes.
If you have a system which flows by itself,
Yes.
how do you find what was the shape of the box?
It was very large, right?
It was like if you put them in it's weird to have something which flows by itself and looks like
Yeah, I mean, so what we've done actually on rheology is you can take a tool and you can put it at zero shear stress condition, right? And if you put this tool under certain condition, it's just going to spontaneously start rotating in one direction for a while, and then it will switch and it will rotate in the other direction. And so I really think that these things can spontaneously generate macroscopic flows. But then if you do a rheology with classical experiments, you just get a G prime and a G double prime that really looks like a gel, right? So I mean, it's a mystery. Yes,
if you did it strain rate, continuous in one direction, probably get really weird.
Yeah, we haven't done that yet, right? I mean, so this is what we are setting up to do. So I have no deep insight into these kind of experiments, right? So,
But for liquids,
Yeah.
the theory certainly asks for it that it seems to not only flow spontaneously, but also if you actually make sure to do the rheology so that you get an effect in the stuff.
yes. So yeah, no, I mean,
I think it's also strongly dependent in that case if you do the figure because stress.
these experiments, no. So
the rheology of the system has been studied to some extent. Not experimentally.
And so that's something that we want to do is we want to make these things and usually what you do in a normal rheology experiment is you put the force to the boundary and make the fluid flow, but now with these active materials I think you can the fluids can push on the boundaries and produce the force, so you kind of reverse the experiment, and that's where we're trying to go, but I'm not really a rheologist, so it's taking long time. So far what I've talked to you is about isotropic gels that are just randomly oriented, okay? And so what I want to ask next is what happens if I increase the concentration of the filaments. What I'll get is a transition to something that has a long-range orientational order. And so this is, a characteristic. If you have rods, and we do have rods, at low concentration, you'll get an isotropic phase in which all the rods are randomly oriented, and the center of mass of all the rods are randomly oriented, right? And so this is just your classical isotropic phase that's been studied to a great extent, right? If I increase the concentration, you'll undergo a first-order phase transition from an isotropic phase to a nematic phase. In a nematic phase, all the rods point along the same direction, right? And so that means that there's a long-range orientational order. There's an order parameter associated with the orientation of the rods. Okay, but if I look at the center of mass of each of these rods, they would just be isotropic, right? And so that's why this is called a liquid crystal. It has long-range orientational order, but short-range positional order. And then you can go to higher order things like Nematics, but that's not really important for my talk. And so what's going to turn out, this is a perfect nematic, right? So these things are perfectly aligned, but if you just take your nematic and if you put it under cross polarizers, you'll often have defects that you try to fight against. And the other thing that's very important to point is the symmetry of equilibrium nematics is such that molecules will often have a head and a tail, but these molecules are equally likely to point up and down, right? So what you describe it is you don't describe it by arrows, but you describe it by something that is a quadrupolar order, right? And so this director that describes the average orientation of the molecules doesn't have a head and a tail. And so that's the fundamental symmetry of the system. And so, as most things in nature, these nematics have defects. And this is one type of defect that they can have, and I guess you'll hear much more about it from Mark in a few days. But so here I have a nematic arrangement, and I have a core of a defect, right? So I can define the average alignment of the molecules everywhere but at this core, right? So I can't tell, if I sit at this core, I can't tell what is my average orientation of the molecules. It's similar to like sitting on the North Pole and asking what is the direction of the wind, right? North, east, south, and west have no particular meaning. And so you can have these topological defects, and the way you define a topological charge is to take a test rod and complete a circle. And if you complete a circle, if this rod rotates by 360 degrees, you get a topological charge of 1, right? So this rod rotated by 360 degrees, and so the topological charge of this defect is 1. Now what I told you is that the nematics do not have an up-down symmetry, right? So so you can also rotate a rod by 180 degrees, and you end up at the same position. And that's so you can actually have lower energy defects that are shown over here. And so in this defect, if I take this test rod, and if I go around, actually this is in the wrong direction, but if I take this rod or this rod, and if I go around and rotate it along the director, this is only going to rotate by 180 degrees, not 360 degrees. So this kind of defects have a topological charge of +1/2 and a -1/2. Okay? Any questions from students about this, or you learn much more about it from Mark, I presume? Yes?
Can you get a -1?
Yeah, right. Yes. So if you just take your nematic liquid crystal, and if you look at it under polarizing microscope, you will see something like this, okay? And so this is a polarizing microscope, and polarizing microscope tells you what is the average orientation of the molecules, right? In a nematic liquid crystal, what you have is you have structural anisotropy, right? These molecules point, the structure is different in this direction and this direction. And that means that the index of refraction is different in this direction and this direction, right? And so if you have different index of refraction in two directions, you'll get what's called a birefringence. And so if you put things in between polarizing microscopes, you'll see this birefringence, and that's shown over here, okay? So you have these defects, and by looking at these poles, you can say whether these defects are of charge 1/2 or 1. So you can see that a defect of charge 1/2 has 2 lobes, and a defect of charge 1 has 4 lobes. These things are usually bad news, right? If you had this on your laptop, I presume your laptop LCD wouldn't work very well. And so the trick, the whole reason why nematics are as useful as they are is because you have these giant rubbing machines that rub the surface of your LCD display, and that assumes that gets rid of the defects because it forces the molecules to point in one direction, right? And so this is an image of such a rubbing machine that is in some factory, probably in Korea or Taiwan. Okay, so we can take our microtubules and we can make nematic liquid crystals by just increasing the concentration. We can't do it in 3D, but we can do it in 2D, and we are trying to do it in 3D. But 2D is actually easier to visualize and things like that. So you have an oil at the bottom, you have a water on the top, and you have your microtubule suspension in the water surface. And so you have an oil-water interface, and this oil-water interface is covered with a surfactant. And so the surfactant prevents the proteins from attaching to the oil and denaturing this oil. And so what you can do is, again, this is an image, and here I'm increasing the microtubule concentration in the water phase. And so the water is below the screen, the water is above the screen, and the microtubules just get absorbed onto the oil-water interface due to the depletion interaction. And so this is, here at very low microtubule concentration, I see individual filaments, right? At intermediate microtubule concentration, as you get bundles, and these bundles, as I increase the microtubule concentration, grow in thickness, and eventually you get something that really looks like a nematic liquid crystal, right? So this is a dense array of these bundles, and here I have my, topological defects of -1/2 and +1/2. And so again, this is, just to remind you, this is a topological defect of +1/2, and you can really see you can directly visualize it. And this is a topological defect of -1/2. Okay? And so, but this is driven, right? So there's motors everywhere. And so unlike a conventional nematic liquid crystal that just in equilibrium molecules are just sitting there, you have a bunch of motors that come and push the filaments apart from each other. And so what you get is this kind of, this is just a larger scale. You get these things, they do very unusual things, right? So this is your active nematic liquid crystal, right? So this is its dynamics, and you can directly see it. And so you can see these defects, and the whole thing exhibits large-scale flows, okay? And so you can see this defect travels, this defect is traveling, and at some point these defects are being generated and annihilated. I don't think they come here at the right angle, so they just pass one another, but there should be an annihilation event somewhere. Here there's a pair of defects being generated, and here you have an annihilation event. So you started with +1/2, -1/2, and a +1/2, and two -1/2s annihilated. Here you have this, again, a spontaneous generation of defects. So did you see it was a uniformly aligned state, and now I have a +1/2 and a -1/2 state. And so the challenge is to really describe, this in kind of quantitative detail, and that's what, Christina might talk to you much more about. So in a certain way, this is very similar to what's happening with the isotropic gels. For the same reasons, these active nematics have a bend instability. And so, for example, here you see an active a bend fluctuation, and this bend fluctuation grows in amplitude, right? And so here the nematic liquid crystal is really highly strained, and you can only bend the nematic liquid crystal so much before you actually fracture the material. And so here, once it's bent sufficiently, you get this fracture line, and this fracture line, you can't define a nematic director here. And this fracture line terminates with two defects. One has a charge of +1/2 and a -1/2. And so what's happening is these defects, you have this bend instability, the bend instability grows, the amplitude, the curvature increases, you get a fracture, the fracture is terminated by two defects, and then this fracture heals, and you generated two defects from something that was initially perfectly uniform, right? Now, if this was the only mechanism, then you would just be generating defects and you would get in trouble. So the other thing that happens in the system is you get a defect annihilation, right? And in a steady state, you have the same rate of defect generation and defect annihilation. And so here you have two defects. They don't meet each other at the right direction, and so they just pass one another, but you'll see two defects at a later point annihilate with one another, as is shown over here. In a second, it should appear. So this is an old sample, and there's some tracer beads here. But here you have one defect coming in this direction and another defect coming in this direction, and you can see that they come at a right angle and they annihilate. And we are trying to, gather extensive statistics to understand, these kind of dynamics, and at what point do they really, what determines the point at which they will annihilate. This also nicely illustrates the dynamics of these active liquid crystals. Is what you've done here is it's just an active nematic, and you take a laser and you shine a bleach spot, right? And so these microtubules over here, there's still microtubules here, but these microtubules are invisible. And so you ask, what is going to happen to these microtubules as a function of time? So you bleach a spot, and then you let your active nematics run, and you nicely see this extensile nature of these microtubules, and they're essentially just becoming longer and longer, and you can see this drop becomes thinner and thinner until no longer you can see it, right? So this is really like, kneading a dough, right? As you stretch, and then you fold, and then you stretch, and then you fold. I think, I've started reading, there's this field of chaotic advection that people have studied for a long time. And so this, if you take those movies and these movies, they look exactly the same. But, here it's internally driven. In convective advection, you need to have random flows. And so we hope to be able to extract some kind of meaningful parameters from this kind of frat experiment. So this is a large-scale movie. This is 2.5 millimeters. You take polarization, and I can define a location of every defect, right? So this is this blue dot indicates a defect of +1 of -1/2, and the red dot indicates a defect of +1/2, right? And so as you let this thing run, you can see that the red defects move along, they exist, they have a fleeting life existence, they're created, they live for about 30 seconds, and then they meet their oppositely charged pair, and then they die, okay? And so we can now do this tracking on large scale, and we can ask questions about what is the dynamics of these defects and how do we quantify it. And so this is just a type of experiment that we can do. So what you do is, here you look at the speed of a defect, okay? And so this is the probability of a +1/2 defect, and you can see it has a speed of about 5 to 10 microns per second, right? And the system is perfectly tunable, right? The speed of -1/2 defect is actually much smaller. And this is at 142 micromolar. I can increase its speed, I can increase its ATP concentration, and the speed becomes much larger, right? At saturating ATP concentration, they move more than 10 micrometers per second. I'm having trouble with my units in Brazil. And again, here they speed up, okay? And so this is related to so you can measure the mean square displacement of these defects, and you can see that the +1/2 defects move much faster. And the reason for that is that the +1/2 defect has this anisotropy, right? So the stresses don't add to 0, and so the +1/2 defect is actually propelled. A -1/2 defect has a spherical asymmetry, and so what's happening is that the -1/2 defects are just being pushed around by +1/2 defects. And so at short time scales, they're pushed, they experience kicks by +1/2 defects, so they move ballistically. But on longer time scale, they move diffusively, and a +1/2 defect moves, much more ballistically, and this is the slope too. Okay. So so you have this. You can think of these defects as particles, right? And so this actually started because Sriram said, well, for a number of meetings, he said, do these things exhibit any long-range order? And I said, of course they don't exhibit any long-range order. You can see that these defects just, it's a turbulent-like flow. And then I asked student eventually after Sriram asked me questions 3-4 times. Well, can we measure, the orientation of these particles? And the student did that, and you get a surprising result, right? These defects now organize into a liquid crystalline state themselves, right? So if I look at the average orientation of these defects, it's actually highly anisotropic, right? And so this tells me what is the probability of a red defect pointing in this direction, and what is the probability of a red defect pointing in this direction. And so you can clearly see that these defects themselves form a liquid crystals. And so that frequently happens in condensed matter physics. I can take defects and I can make a new kind of phase, right? Like a TG twisted grain boundary phase is a phase that's organized by spatial orientation of the defects. Here you have this kind of nematic phase that's organized, it's a dynamical nematic phase, and it's determined by the spatial orientation of the defects. And so we were interested, well, is this just a fluctuation, or is it really true phase? And so, so I can map out an average alignment of these molecules, and I can look at this angle, and I can look at it over, hours, even days, right? And so you have this average angle, and you can see it's, 1.5 radians, and it fluctuates about, but it's pretty steady, right? And so that means that these rods are being created, and they are being created with a specific orientation, and then they move along a certain direction, and then they die, right? So it's not that they are isotropically oriented. And so this is for one specific spot, and then we wanted to see whether this propagates through an entire sample. And so we've made techniques to make these liquid crystals centimeter size, and this is what you see, right? So this is the average. So this is X and the Y position. This is in microns, right? So this is 10,000 microns, which is which is a centimeter, right? Am I correct here? So this is a centimeter by millimeter sample, right? And we've done it on larger samples even. And so this average alignment of the defects, and you can see that it propagates through an entire sample, right? And so this really is, a dynamical nematic phase of liquid crystals that preferentially move in one direction, right?
So the underlying nematic is not a line. So there isn't a coupling between the nematic, defect nematic, and the underlying nematic?
I think yes, there should be, right? So if I measure local orientational distribution function, it's actually going to become long range, right? Because the defects determine what is there. Yes. And so, but this is literally from last week data. So yes.
Sarah, so if you have a defect orientation like this,
Yes.
then does the nematic around it reflect exactly the orientation, or is it up to like a third of whatever it is? Could it be that the reason you don't see long orders?
We haven't taken like a pole scope data and measured the orientational or distribution. We've just measured the movement of the defects, right? But I think if the defects move in a certain direction, that says that the nematic actually has to be aligned. But I'm not sure. I haven't really thought about it all that carefully.
Also, does the average mean there's an average defect density?
Yes.
Does that depend on the ATP?
Not so much. No, I mean things just speed up or slow down with ATP.
So what sense?
Christina?
It's not something that we really understand.
Okay.
It's so basically just to measure the mean number of defects in the system. Initially, it grows with activity, so we grow with the ATP concentration, but eventually it actually saturates. And we don't quite understand yet the landscape. But I'm going to say something about this.
Okay, so
It sounds like magic.
Yeah,
It doesn't depend on it.
it's pretty flat, right? So I mean,
Can I make comments?
yes, you can, of course.
Well,
If you take a random field,
Okay.
like an idle amount,
Yes.
and then from that random field which is generated as a Gaussian random
Yes,
function,
yes.
you draw the principal line of curvature.
Yes.
There's also the field which will have a certain number of defects, which are exactly of nematic type, and they will be there not for reasonable energy. They will be just there because, in the same way as I can tell you a high field amount, which is
Yeah, okay.
Gaussian, can have a certain number of maximum minima.
but then here the dynamics is so it's going to show that we are in the last correlation factor. That there was a landscape that we see to depend on activity. Now, if you put it on physics correlation factor, that landscape depends on activity and seems to correlate with the mean separation between defects. So it's very strange to think about it. The last vorticity, you have literally kind of landscapes associated with that. But actually, in two-dimensional programs, not three-dimensional programs, two-dimensional programs where things are driven uniformly that way, the last thing vorticity is very different ways. So, I mean, I'm just saying. It's not
Well, maybe
something
we
that
can
Okay, let's
discuss this.
Sorry.
No, it's okay. But this is unusual, right? Because you have this mechanism that destabilizes the nematic and creates these defects, but these defects then at a higher level find themselves a way to organize into nematic.
These things are surprising because they depend on the
Yeah. So I mean,
And so central particles.
yeah,
they
sure,
have
sure.
hydrodynamic interactions
Yes.
and all that.
Yeah. I mean, nothing is.
Cannot. They cannot align.
Just one very quick question. The fact that those defects are aligned means
Yes.
there's a mean flow, or is the mean flow still?
No, they're equally likely to go up or down, so they still have a nematic symmetry, right? All right, so as you've seen from this conversation, 2D active nematics are very complicated, right? You have the spontaneous flows, and even the experts don't understand them yet fully. You have this motile defects, and it's this dense soup of defects that interacts via these long-range hydrodynamic interactions. They move with preferential. They exist for a finite time. They created, they die, and so it's a complicated thing to model, right? And so what we wanted to do is we wanted to make something simpler. Like, can you make a system where you just create one or two defects and study it? And so the way we did that is we confined active liquid crystals on topological surfaces. Okay? And so this is, imagine that I have a disk and I want to tile my disk with a nematic liquid crystal, right? I can't do that without creating a defect. If this is my average alignment direction of microtubules, I can tile microtubules like this, or I could tile them in this direction, but I need to have a total topological charge of +1, right? Now if I take this thing, I can just fold it into a hemisphere and I can make another hemisphere, and then I can bring these two hemispheres together, and then I have two defects at the poles that have a total topological charge of +2, and that's shown over here. Okay, so Paul had his Roger Penrose story, so I was motivated last night to do my Roger Penrose story. And so this is Penrose Senior again, and so he was a surgeon or he was in a medical field. And so he wrote this paper about dermatoglyphic topology, right? And so he here says, well, look at the hand, and you have all these defects, and he says direction of the ridges turns by an angle 180, that's the loop pattern, and the center of tetraradius is 120 degrees, and that's this pattern, right? Triradius, right? And so he wrote this paper in Nature in 1965, and then he also did, so there's a lot of medical mouth for, so he formulated topological laws for this hand, and then he went into, people who have, genetic defects, how the topological, if you have more digits or less digits. And so then, so it's an interesting paper to read, and then his son came along and said, well, I can do this much more rigorously, and so he wrote a paper, The Topology of Ridge Systems, and so this was published in Annals of Human Genetics, right? But it's like a completely topology paper, so.
Well, no, the great thing about these papers, because I use these for...
Oh, really? Okay.
He shows that the number of triradii you have in
Yeah.
your hand
Yeah.
is the number of fingers.
Yes.
Yes. And then they do the experiment.
Okay, yeah, right, okay. Anyways, I stumbled on this recently, so, but I guess Paul knew about it before me. Okay, so if you have these defects in equilibrium, you can have a defect of charge 1, and I can take this and I can just split it into 2 1/2s, right? And this costs me no energy. I mean, I actually, you might gain some energy, right? And so on a sphere, I told you that you need to have 2 + 1/2 defects. There's 2 + 1/2 defects, but these defects on the poles can actually split into 4 + 1/2 defects. And you get this, since these defects repel, they'll sit at the corners of tetrahedra. And this was, first done by Jacques Pro and Lubensky, and then Nelson followed it up. And then Vincenzo have actually done experiments on nematic emulsions where they visualize these defects and have shown that under certain conditions they really sit at the corners of tetrahedra, but the thing is much more complicated because it depends on the elastic constants, and people use approximation, and then Mark Bowick has done simulations that show that there's other possibilities possible, right? And so what we do now is we take a dome, right? So we have a liquid dome. Below it is a oil. Above it is a nematic liquid crystal. A liquid crystal, and water is above it, right? So you have this dome. It's coated with an active nematic, and so you can just count the defects. And so you have a +1/2 defect, +1/2 defect, +1/2 defect, and a -1/2 defect. So that has to add up to 0, right? And the topology in a certain way doesn't care whether something is equilibrium or not. You have to always obey it. And so the interesting thing about this is if you actually look at its dynamics, what happens to this thing is it actually just spontaneously circulates in one direction, okay? So unlike a flat two-dimensional nematic that is totally chaotic, what you have here is you have a circular flows, and it can go in either left-handed or right-handed direction, so it's spontaneously broken symmetry, and this thing is going to circulate for multiple hours, right? There's a lot of energy here, and you just circulate around, and so and at some point, you might be having only 2 +1/2 defects, or you might generate an additional pair, and so that's something that we are trying to quantitatively understand, and I think you can change ATP concentration, and you can see how this emergence of this circular pattern evolves as a function of time. And then one could also ask is if I bring 2 of these rotators, can they synchronize if I bring them together? But what we've actually made more progress on is by encapsulating these things into spherical shells, okay? So you have these nematics, and if the sphere is smaller than your characteristic buckling size, they'll just be they'll be in the bulk. But if you make the shell of the characteristic buckling size, what happens is the 2 filaments are pushed apart, and then they buckle, and they are pushed on the surface, right? And once they're on the surface, there's no way for them to escape into the bulk, because escape into the bulk requires contraction of the material. So they're being pushed on the surface, and they're constantly pushing outwards. And so you get this very nice nematic shell that's shown over here, okay? So this is actually a vesicle that's viewed from the top and the bottom. And so you see 3 defects. So this is a vesicle. I can view it from the top and the bottom. And these are the views from the 4 sides, okay? And so there's 4 defects, and there's no additional defects. And actually, for this sphere size, you will only get 4 defects. And these defects have an infinite lifetime, right? Because there's no -1/2 defects to annihilate. And so they're just moving around they're just confined to eternal existence on this spherical surface. And so the interesting thing is, again, these defects are motile, right? And so they're being propelled on the surface, and you can see how they move on the surface. This is a spherical shell, and there's elastic forces that causes them to repel each other. So you can see, for example, here, this defect moves in this direction, this defect moves in this direction, and they repel one another, right? Yes?
How do you do the experiment?
How do I do the experiment? I don't do the experiment. I tried to do these experiments, but it was a terrible failure. And so when I went to Munich, so my students followed me, and they made it work, right? So. Okay, so yeah, I think I know how they do it. So what is your question? You just have you put your active mixtures into a vesicle, right? And they will just spontaneously coat the surface of the vesicle with a nematic.
The vesicle has like also something at the surface. It's not a solution.
No, this is a vesicle.
So you have a lipid.
Yeah, there's a lipid on the outside, and on the inner leaflet of the lipid is coated with active nematic.
And what's inside that? Nematic or what?
No, it's empty in the middle. So it's a single monolayer, or
So it spontaneously goes.
yeah, it all goes to the surface, and yes, yes, yes.
You said they interact elastically,
I mean, the defects there is going to be elastic interactions between defects.
but is it really, should I think of it really like it's sort of stretching and elastic, or more like a constraint? Like because it looks like they kind of approach each other at particular distances and they rotate away.
Yeah, because I mean, the elastic interaction must increase. I mean, it's logarithmic, right? Or is that correct? If I take 2 defects on the surface, it's a long.
One of the things that bothers me here is all the statements about being logarithmic refer to the fact that there will be an energy that you want to make nice.
Yeah, okay, yeah.
Whereas I really think that here the stuff is there for kinetic reasons that are, at least on this length scale. See what I like that? You get an logarithm by minimizing an elastic energy.
Yes.
So the medium, in order to have elastic energy, the medium must be all of them out. And this system doesn't satisfy that.
Yes. Yes.
Derivatives would be the
Yes.
forces of the motion.
But I mean,
On very short lengths, on very short.
I will talk about this later, but I will give example.
Yeah.
In short, if you describe these things as something like particles, you can do it by
Christina, actually, you can reproduce fairly decently, by just modeling these things as particles that move, that are self-propelled particles, and they have repulsive interaction.
Right, okay. Which is a different
But so the system is inherently frustrated, right? Because this defect in equilibrium just sit at the corners of tetrahedra, but now they want to sit at the corners of tetrahedra. They want to be also propelled, right? They want to acquire finite velocity, as you can't really, you can't rotate a tetrahedra where all the points move at the same velocity with the same preferred direction. And so the system is inherently frustrated. And so what we've done is we've actually tracked the position of all the beads, of all the defects as a function of time, and you can see as they're diffusing on the surface over here, right? And so you can watch this for a while, and so the way we quantify the position of these defects is, for example, imagine that this is my tetrahedral configuration, my defects are the corners of tetrahedra, and then I measure the angle, I measure 6 angles. Angle alpha 23 is the angle between this point and the center of the radius, right? And so all of these angles in the tetrahedra are going to be same, of 109 degree. On the other hand, if, for example, I have a great circle inscribed into a sphere, I'll have 4 points, right? And the angle alpha 12 is this angle, which is going to be 90, and the angle 13 is going to be 180, okay? And so I can measure these angles experimentally because I know everything about my system. And if I do that, I mean, I can visualize everything about my system. I don't know much about the system. So here I plot the 3 angles as a function of time. And what you can see is that they actually exhibit, so maybe it's more informative to look at this sample, this average value of the angle. And so you'll see periodic oscillations, right? So these things really oscillate between 2 states. And so one state is indicated with the blue line, which is 109 degrees. And so this is the state that's a tetrahedral state. And then it almost reaches the state of 120 degrees, which is a state where all the defects are lying in the same plane. And so you can see these oscillations also in this chymogram, where these 2 defects light up, and then these 2 defects light up. These 2 angles light up, which means that they become larger, and then it goes so on as a function of time. And so, you can also plot the distribution of the defects at the extrema, and so in the blue, it's again something that's close to the tetrahedral configuration, and in the red, at this maxima, you get a number of angles that's around 90 degrees, and then some angles that are 180 degrees. Yes? Yeah, but again, we have this ATP regeneration system, and so you can just slow these things up. And actually, what limits, how long these things exist is, well, vesicles are floppy, so vesicles die pretty quickly. But emulsions, what limits is proteins in nature, right? So you're constantly replacing proteins in your body that are no longer functional. But we don't do that in our system.
If you let them run out of fuel, you just see the tetrahedron in a fixed configuration?
We haven't done that in this case yet, because actually the vesicles die faster than the ATP runs out. But we could do that in a different configuration. We haven't done that yet, but that would be a good experiment. And so you can take a Fourier transform, and there's a specific frequency. Okay, so vesicles are floppy, right? So floppy vesicles, that means that the forces exerted by these defects could, in principle, deform the vesicle configuration. So what we do here is we have an active vesicle. It's just a two-dimensional projection. And what you do is you add a lot of sugar to the outside. What that happens is the osmotic pressure on the inside is different than the osmotic pressure on the outside. And the water is going to flow out. And at that point, the vesicle becomes very, flabby and flaccid. And so there's a lot of extra surface. And so here we start, with a very tense vesicle. It's essentially spherical. And then, the water is rushing out. And as the water rushes out, this vesicle starts to deform. And as it deforms, it grows this kind of protrusions, right? And so here you see 4 protrusions. And as it becomes more and more floppy, these protrusions grow in size, and so this really looks like something that you've fished out of your pond, but it's really a material that has just 3 components. And the interesting thing is that the topology determines that at any given point you can only have 4 of these deformations, right?
Why can you have 4 at the end?
And for these curvatures, you don't get a creation of an extra pair, right? And so I think if you go to larger vesicles, at some point, at some curvatures, you will start generating more defects. But we haven't, we haven't been able to explore that regime. And so this is the 3D reconstruction of these vesicles, right? So you have something that's spherical, it becomes oblong, and then you start growing these protrusions. And then if you start decreasing the osmotic pressure, it goes back to a spherical state. Okay, so I'm almost done. This is the last thing I want to talk about, is these are emulsions. So now what we do is instead of encoding it into a vesicle, we actually put these things into an emulsion, right? The advantage of an emulsion is that the surface tension is much weaker. The vesicles will actually, fall apart after half an hour to an hour. But the emulsions last for a long time. And so what you can see is this is, again, an emulsion that has the surface nematic. And if you look at this emulsion, it actually acquires motility, right? So you have this internal flows, and so you have an internal flow going in this direction, and the emulsion goes in this direction. And so the way we do that is we have to increase the friction between the surfaces, and so we slightly confine the 2 emulsions. So so it's really this kind of geometry shown over here, okay? And so,
Why do you need that?
huh?
Why do you need, because here you said that you need, to be able to push on the water.
Yeah, I think you need to increase the friction between the vesicle and the surface of the wall, right? If you just have a thing, on the surface, it's not going to move very far because it doesn't couple too strongly hydrodynamically to the wall.
But if you are in bulk, and you say you move inside and you break the symmetry, you could imagine that you get slippage and then the water should move through. Like in the same way that you have streaming or cilia. Could you imagine this to move in 3D?
You mean away from the surface?
Yes, like the CDAT.
I don't think it would swim by itself because I think you need to somehow break the symmetry, right?
Spontaneously done.
Yeah. So we're actually setting up to do a donut experiment. Okay, so you have these, so you can just generate these things using microfluidic devices. So these are huge emulsion droplets, right? This is 100 microns in diameter, right? It's totally non-Brownian. And so most emulsions of this size would just sit there as bricks, but these emulsions actually, acquire biological functionality, and they can crawl around on the surface, right? And so we started measuring the mean square displacement of these emulsions, and what you can see is the mean square displacement is shown over here, okay? So at short time scale, it's ballistic. And we believe that that's related to, this is on time scales less than 10 seconds. So this is related to how long it takes for one defect to go across the surface, okay? And then on longer time scale, it becomes diffusive, right? Because these defects, as they come to the surface, are uncorrelated. And so you get a mean square displacement that looks like this, and we are trying to, come up with some kind of functional forms that would describe the motility of these droplets. And then, the nice thing about these droplets is that everything that you need for this droplet motility is encapsulated within the droplet. So you could, make a mixture of fast and slow droplets or things like that. And so, this is a preliminary experiment where we just take a lot of droplets and, we confine it. And so, the question is, if you have something like this, how would this crystallize? Does it crystallize? At what concentration it crystallizes? But the thing that we are fighting against is the drift, right? And so that's actually very non-trivial to make, a non-drifting sample. But hopefully we get there. And so you can see that they do interesting dynamics, right? And so we want to study the dynamics of such crystals. And this is again going to last for multiple days. Okay, so this I'm at the end, right? So I've started with something that's very, very simple. It's a microtubule that's extensile. And so what we hope to do in the future is use the techniques I've described yesterday to really measure the mechanics of the single microtubules, to measure the forces generated by these motors and things along those lines. And then, we've just, you start increasing the complexity, and you don't need to go very far to obtain something that looks like a biology, right? So we made these gels, active liquid crystals. By confining it onto a sphere, you make a robust oscillator, and so this is kind of my, so that's where we are going, right? And the question is, can you actually, can you make some materials that are useful, but also can you compare it quantitatively with the experiments? Now, if I talked about friction yesterday, so if you increase the friction, you get something totally different, right? And so, for example, here you'll get these clusters, and so you'll get this cluster over here, and then these clusters at some point just become unstable, and they explode, and they do all this kind of very crazy dynamics. So the phase space is, you make microtubules longer, or you increase the PEG concentration to much higher values, right? And so instead of a system being extensile, at some point it becomes contractile, and it forms this kind of structures. And so, for example, here you can have one of these stars that's collapsing on itself, and then it spews microtubules, and these microtubules become lodged at this air-water interface. And then if you focus on this air-water interface, that's zoomed in over here, is you'll see that these lodged microtubules over here start exhibiting very interesting dynamics, right? And so they spontaneously organize into these, because they are lodged now, they can no longer extend, and they start beating. And so, and so you can create, these kind of, if you take these extensile bundles and you lock them at their base, they can no longer extend, and they start beating, right? And so in such a way, you've really recreated something that looks like a biological spermatozoa. But, biological spermatozoa has 650 components. This has 3 components, right? And so, I'm not saying that this has anything to do with biology, but, it's an interesting, instability of these bundles towards beating. And so with that, I'd finish. And thank you for your attention. Yes.
Wonderful talk. I just wanted to ask you to bring you back to this question of the magic number that Paul was asking. Can you plot the density of the defects just qualitatively as a function of time? Tell us what is the density once you reach the steady state.
Yeah, I can do that. I have plot somewhere. I can email my student and I'll give you the plot. It increases slightly over time.
And it's saturated.
It saturates.
It's saturated at a value which is independent of the activity, but presumably...
We are doing those experiments quantitatively now to really see how, first, qualitatively doesn't depend on ATP concentration at all. The only thing that ATP does, but there might be a lower, if I...
presumably the time it reaches the duration will depend on the...
Yes, sure. No, that's the nice thing about our system is that we constantly regenerate ATP using, enzymes, and so as soon as you get ADP, the enzyme regenerates ATP from another source. And so the dynamics is truly steady state, right? So that makes, otherwise if that, all of this would die within 15 minutes or so.
The enzymes makes them homogeneous.
Yeah, they're just enzymes floating around, the same enzymes you have in your cell, right? If that you used to. Yeah. Yes.
You think to repeat these experiments in a cover surface, another cover surface, right?
So something that we really don't understand is we can't reproduce this on a solid interface. We can't get a liquid crystal, active liquid crystal on a solid interface. We make it chemically identical to a liquid-liquid interface, but the microtubules do not absorb onto the solid interface. It's as though they are being repelled from a solid interface. And so that makes any kind of topology more difficult, right? So we need to have a liquid-liquid interface. But
I think in the planar geometry, the total topological charge must be zero. Okay. Maybe
well, it depends on your boundary conditions, right? So
in infinity.
Yeah.
But maybe this is the reason for the charge, the defects may born in pairs. No.
yeah, I mean you have to have
Cover surface is the defects are part of the equilibrium system. Maybe.
Yeah. I mean, yeah, but yes, exactly. I think you're saying the right thing. I can't have on a curved surface. I need to have defects, right? Just by topology, and so I need to have 4 defects, and so they are there by default, and so that's But we would really like to create a -1/2, but that's not obvious how to do that. Okay, I guess we go to the mountain, right? Burn some ATP.